Hello dear learners, in our today's class we will learn about some of the important cloning and the insertion vectors which are popularly used in the biotechnology. So our syllabus mentions about 5 to 6 important vectors and we have to study about them one by one. So we'll start first with the vector Lambda Bacteriophage. So here I have presented to you the genomic diagram of the bacteriophage lambda vector. Along with this diagram, I'll also be explaining about that what are the important functions and how does the vector works, means what is its main mechanism, that how is it working and how will it transfer the gene of interest into the host. So we all know that Lambda bacteriophage is a kind of temperate bacteriophage, and this is the bacteriophage of E. coli, and it can also be used as the cloning vector. So basically, this Lambda bacteriophage, as we know what is the function of bacteriophage, is that it will try to transfer its genome into the E. coli or into any particular bacteria. So while it is transferring its genome into that bacteria, we will insert our gene of interest into this genome so that this gene of interest can also be transferred into the bacteria. Now the process of transferring the genome of bacteriophage lambda is very efficient, and we are exploiting this efficiency of bacteriophage lambda so that our work can also be done simultaneously. Generally, the lambda phage contains a linear double-stranded DNA. This linear double-stranded DNA contains cos sites also as it at its ends. Now here it has been presented to you in the circular form because let's say if this is our bacteriophage and once it is infecting the particular bacteria, so the linear DNA consists of certain kind of cos sites which I'll probably label in pink color. So suppose here are the cos sites. Now once it is entering inside the host, the cos sites will help to circularize this entire DNA. Means cos sites will basically produce a kind of sticky ends, and these sticky ends will help to circularize this entire DNA. And why the circularization is important, so that it does not face any kind of stress inside the host, so that enzymes cannot be perfectly acting upon this, so that it is not prone to degradation. Considering all of these reasons, the circularization of this DNA is very important. After that, it will be trying to interfere with the host genome and the machinery. So our main aim would be that we have to take this cos sites from this bacteriophage, and we'll study it also in another important topic, which is perhaps the cosmid. In that cosmid, we'll understand that we are taking a normal plasmid, we are inserting the gene of interest in the normal plasmid, and also we will be taking the cos sites, and these cos sites will be derived from the bacteriophage lambda. This will be derived from the plasmid, and this will be our gene of interest. Combining all these things also, the product which is there, that is cosmid, proves to be very efficient. But when we talk about bacteriophage lambda, although it is not that much efficient in process to transfer our gene of interest, but yes, because it was a foundational approach, so we also need to study this. Now one must know that the bacteriophage lambda consists of 48,000 base pairs, means its genome is approximately 48 kilobases. Now it consists of certain genes which are called as replication genes, whose replication has to be done. There are certain lysis genes. Once the genome enters inside the host, it has to do the lysis of the host. That is why the lysis genes are important. The coat genes are important because it will help to secrete the coat proteins, and these coat proteins are important while the genome is in the bacteriophage itself. Now there are some of the very essential genes also. Then there is a green fluorophore protein which we can insert, and this green fluorophore protein will help in the detection whether this particular genome has entered into the host or not. Then there is an origin site, and then there are these restriction sites. Restriction sites are basically the multiple cloning sites, means in that particular site there are multiple particular area, there are the multiple uh, restriction sites present of different enzymes. For example, eco r1 can also act here. For example, bam h1 can also act here, depending upon There are different sites, and you can act any one of them. Like you can cut from there, and then you can also insert your gene of interest. And finally, because it consists of cos sites, these cos sites are important to circularize the entire DNA. Now, once the recombinant phage has been produced by inserting the gene of interest in the multiple cloning site region, now this has to be transferred into the host. Now, because we have isolated this genome, we cannot do this inside the head of the bacteriophage. So firstly, we have to isolate this, then we have to add our gene of interest, and now this has to be reinserted into the head or the capsid of the bacteriophage. Once it has been done, after that this will be transferred into the host, and inside the host it will undergo circularization because of the presence of the cos sites. So the normal process can be happening. Now the insert capacity of the bacteriophage lambda is approximately 20 kilobases. 20 kilobases simply refers to that we are adding approximately 20 kilobases extra in the multiple cloning site. So here, although the total base length or the total thing is of 40 kilobases, so still we can add 20 kilobase extra, that is 20 kilobase gene of interest. So this is its maximum carrying capacity. If you are adding more than this, then the process will not be that much efficient, because then the gene of interest will be prone to degradation. So efficiency is maintained only up to 20 kilobases. Now, what are the advantages of using lambda phage? So one of them is that because it has a very high infection efficiency, so it can easily transfer your gene of interest into the host. Secondly, it can be used to construct the genomic libraries. Means here you can insert your gene of interest and you can preserve it for later use. Also, one thing important is that once this bacteriophage lambda is infecting its host, it will lead to the formation of plaque. And once the plaques are formed, you can easily identify that yes, this host consists this lambda bacteriophage genome. So all the transformed colonies are those colonies which have taken up the foreign DNA or which have taken up the bacteriophage DNA. The transformed colonies will have to form the plaque, and through this plaque you can easily detect that yes, these colonies are transformed and these colonies basically contain the foreign DNA or the gene of interest. The major limitation can be that it has the packing size limitation. It can only pack up to 20 kilobases extra, not more than that. So this is all about the bacteriophage lambda. Now we will move on to the next important vector, that is M13. Now this M13 is again an important vector, and we say that it is a kind of a filamentous phage. This filamentous phage, firstly, it is 900 nanometers long and 9 nanometers broad or wide. Now the insert capacity of the filamentous phage is very less. You can say approximately 1 kilobase only. So if you are inserting anything greater than 1 kilobase, you cannot clone it and you cannot store it, because it is prone to degradation. And the efficiency of this M13 phage, this M13 is also sometimes referred to as F1. F1 simply refers here to filamentous phage. So the efficiency of these phages decrease a lot if you are inserting your gene of interest greater than 1 kilobase simply. So yes, this M13 phage is also very important. Along with that, if we talk about the diagram which has been shown here, so it consists of the ori site. This ori site is the place from where the origin will take place, or the origin of replication will take place. The entire genome size of the phage comes out to be 6.4 kilobases. If we compare it with the bacteriophage, which had 64 kilobase, sorry, 48 kilobase of the entire size, here it is only 6.4. The insert capacity of the bacteriophage was approximately, lambda bacteriophage was approximately 20 kilobase, but the insert capacity of M13 is approximately 1 kilobase. Now the M13 phage is also very important, because here it has a major function to produce a single stranded DNA. Once this is entering inside the host, although initially it is single stranded only, so we can say that this M13 is initially single stranded DNA. then it will enter inside the host. Inside the host it will convert into a double-stranded form. Now this double-stranded form will do the process of replication, and this double-stranded form will very much look like a plasmid. Now this double-stranded form of replication, which we also call as DSRF, it can produce two things. One is it can produce the double-stranded daughters, and if needed it can also produce a single-stranded DNA. Now this property we are exploiting, and what do we do is that our gene of interest, whatever the gene of interest that we choose to have, we will insert it during this phase. So when the M13 is in double stranded form, what do we do is that we suppose take one particular restriction site. Let's take it for eco R1. So it will cut here on the particular site of this double stranded replicative form of this M13, and same eco R1 we will also use to cut our gene of interest. Now what we are going to do is, suppose here it has cut at this place and here it has cut at this place. Now we will take this particular gene of interest from here and we'll transfer this here. Once it has entered here, now it will be a recombinant one. So it is recombinant DSRF. Now this recombinant DSRF can produce the double-stranded daughters also, and it can produce single-stranded DNA also. Additional details if you need to know is that initially while it was a single-stranded form, we say it is a positive strand. And once it is in double-stranded form, so one original strand was positive orientation, the another strand formed is in the negative orientation. Now when it will be forming the single-stranded DNA, so this single-stranded DNA can either be in positive orientation and either can be in the negative orientation. Now the gene of interest which you are adding in the double-stranded form, while it will be cloned, now clone means while it will be replicated, it can be either inserted into the positive strand or it can be inserted into the negative strand. So 50% of the strands would be positive and the 50% would be negative. These 50% would contain the gene of interest and these 50% would also contain the gene of interest. So it can depend, it is a total chance event. Also one thing important you need to know is that these M13 phages, once they have to enter inside the host, so they can enter inside the host only through the sex pilus. So there are many of the bacteria which form a pili. Suppose this is a bacteria and this is forming a pili. Now this pili is known as sex pili. Why this pili is formed? Because they have to transfer their plasmid into the another one. So those bacteria which form the sex pili are F positive and the bacteria which accept that are F negative. So basically the M13 will enter only through the sex pili. So we can say indirectly that M13 is only affecting F positive host. It cannot act upon the F negative host. So these are the important points which you need to know. One thing you can also learn is that these M13 vectors sometimes contain LegZ gene, and the LegZ gene is one which will code for, suppose there is the LegZ gene present inside the genome of the M13, it will code for beta galactosidase and this beta galactosidase will act on X-gal, and we have already learned that how it helps in the blue-white screening in the previous semester. So this can be useful to identify the transformants and the non-transformants. The M13 also has very little or approximately you can say negligible non-essential DNA, means every part of this DNA is important for M13. So you cannot remove any part of the DNA by just thinking that if you remove that part and you can add a new gene of interest there. So this cannot be done. While it is possible in other vectors, because other vectors contain much of the DNA which is of no use, so simply you remove those DNA parts and you insert your gene of interest there, but this cannot be done with M13 because most of the genome of M13, and already we have seen it is already very small of 6.4 kilo base only, so it is very essential DNA for the M13 phage. So in the next video we will be concluding with the another vectors also, the other few vectors which are left, and after that we'll complete this topic of vectors.